Hi, today I'm going to present the FYP project, Feature Selection on Transcriptomic Data for Identification of Novel Biomarkers for Bipolar Disorder. Genomic psychiatry is actually a recently expanding field which holds much promise in biomarker discovery for psychiatric disorders. However, the high dimensionality of genomic data and relative small cohort sizes at the outpatient clinic imposes a challenge for clinically significant analysis of transcriptomic data. Salient genes need to be identified for discovering novel biomarkers. With the transcriptomic data from a cohort of lithium-treated bipolar disorder patients, non-lithium-treated patients, and healthy controls, we proposed a pipeline of univariate filtering and multivariate feature selection with machine learning-based approaches. For the univariate filtering stage, we feed the original 55K genes into statistical tests like F-test, T-test, and chi-square test, and evaluate their performance by the prediction accuracy using the selected genes and a decision tree classifier. The genes selected by the optimal filtering method were then used for further feature selection. For the multivariate approach, we used the recursive feature elimination, RFE, with various machine learning models as its core. To make the comparison, nested cross-validation is used to find the optimal set of genes giving the best prediction accuracy. The outer loop is a 10-fold cross-validation for comparing the RFE with different model core. The inner loop is a 5-fold cross-validation for finding the optimal set of selected features for each model core. After the genes are selected by RFE with the optimal model core, they were then used for downstream biological analysis, such as gene set enrichment analysis and gene ontology analysis. We have conducted experiments among these 4 comparison groups. The false positive rate adjusted p-values of 0.05 and 0.01 were used for each test. It is observed that the F-test provides the highest selectivity as well as the best prediction accuracy among the 3 methods for this dataset. This suggests that the variances in gene expressions could be the key differentiating factor in this dataset for different cohorts. Hence, the F-test was selected amongst the other tests as the optimum univariate filtering method. It is also worthwhile to notice that the gene subsets obtained from the method achieved comparable prediction accuracies with the original set of genes. Some even achieved higher prediction accuracies with a significantly reduced feature size. Therefore, the univariate filtering method does help to extract the salient genes that contribute to the classification of different cohorts. Next, we have chosen the 2 comparison groups that still need further feature selection for recursive feature elimination. Nested cross-validation was used to compare the different models used as the RFE core, and logistic regression has shown to have the optimum performances. By applying RFE with logistic regression, we have obtained 292 genes for healthy control versus all bipolar disorder patients, and 1,950 genes for healthy control versus lithium-treated patients, with test prediction accuracy of 93.5% and 93.7% respectively. Comparing the prediction accuracy at different stages of feature selection using logistic regression model, we can see that the pipeline of feature selection on transcriptomic data readily reduced the feature space and improved the test prediction accuracies. The 292 and 1,950 genes selected from the 2 comparison groups were further validated with downstream JSEA and GOA analysis. Here are some results of the top enriched pathways obtained. From these enriched pathways, it is observed that the immune system and its constituent components were activated as indicated by the upregulation of relevant pathways to both the adaptive and innate immune system. It also corresponds to the presence of systemic ongoing low-grade inflammation in the patients of affective and psychotic disorders. It confirms that the bipolar disorder patients, especially those treated with lithium, have activated or dysregulated biological pathways, which point to the pathophysiology of BD and mechanism of potential alleviation and therapy. In conclusion, this study has proposed a feature selection pipeline for discovering novel biomarkers from whole blood transcriptomic data that combines the merits from both univariate filtering with statistical tests and multivariate methods with machine learning models. We conclude that the feature selection pipeline can overcome the challenges of high dimensionality in genomic data and extracting salient features highlighting related biological pathways for downstream analysis. Biological inferences from the genes selected have also validated their potential as novel biomarkers for the pathophysiology of bipolar disorder. Increased knowledge of these pathways have potential to be translated to better therapy options. Thank you.